and in today's class we will discuss the beneficial insects of last 5 years paper. Okay, so everyone join quickly and be active in the chat box so that this session will become interactive. Students, we will continue with the class, and the first question is origin of silk gland in silkworm. Can anyone knows the answer? Origin of silk gland in silkworm. Yes, Chaya and Anchal both gave the answer A, and gaming confidently some person gave 1. Yes, it's correct. So the origin of silk gland in silkworm is ectodermal origin, and here we should know the silk gland structure, and it's important. So quickly I will explain that. Okay. This is somewhat silk gland structure, and here we can see the in silk gland this is the spinneret, and this is Phillips gland. Then after this is the anterior region. This all portion, the widest portion is the middle one. Okay. And this is the posterior region. Here we should know one thing, from where the different proteins were formed. And mainly in silk we will have two proteins. One is the tough elastic protein, that is fibroin, and somewhat gelatinous matrix protein, that is sericin. Okay, for that we need to think. Yes, and it's ectodermal origin. Now we were discussing this structure. Okay. And mainly while coming to the secretion of the different parts of the silk gland, in the posterior region it will secrete the fibroin, and mainly while coming to sericin protein there will be sericin 1, sericin 2 and sericin 3. So different regions of this middle region will secrete that anterior, posterior and the middle part of the silk gland. So here it will secrete the sericin. So it is a gelatinous matrix protein. That thing you should remember. And it is the tough elastic protein. Okay. And while coming to in 2019 paper, they have asked the proportion of this fibroin and sericin protein. So it is usually constitutes the 75%, and usually it constitutes for 25%. And mainly fibroin is the inner protein and sericin is the outer coating protein. And like this, from these two posterior limbs, we will get the two threads of the fibroin protein, and that is called as brims. Okay, and these will be merged into one and coated with the sericin, that is called as a bave. So that is the spinneret, that is median projection in the labium. Median projection in labium, we can see here. So this is the typical silk gland structure. Yes, Anchal Gupta gave one reply, that is fibroin 75% and sericin 25% she gave. Yes, that's true. And here one more thing is that we need to think the two silk threads without sericin protein, that is only with the fibroin protein, is called as a brims, and usually it is coated with the sericin protein and combined into a single thread, that is called as babe, and these two terms are important, brims and babe. Okay. So answer is ectodermal for this. Silk glands in Morbix, Morius, labial glands, this is discussed. And while coming to second question, we can see the given below are two statements. And statement one is royal jelly secreted by the honeybee. And statement two, drones can't sting. So first, while coming to the statement one, can anyone answer this? Yes, Nishan gaming confidently, Chaya gave A. And gaming confidently put one message that इससे और कैसा क्वेश्चन बन सकते हैं. So while coming to the first one, what his doubt is, rest of the how the rest of the questions can be asked. As I explained now, the two fibroin threads can be called as brims, and when coated with the single sericin protein, that is called as a babe, that can be asked, and from where the projection can be come, that is spinneret, and where the fibroin secretion can be done, that is the posterior region, and anterior portion is very thin without any secretion, and that is important. Okay, such questions in statements or some, so choose the correct answers may come. And while coming back to the second question, the statement 2 is royal jelly is secreted by the honeybee. Yes, royal jelly is secreted by the honeybee, that too specifically the worker bees. And drones can't sting. And what is this sting apparatus? Sting apparatus is sting apparatus is nothing but the modified ovipositor. In males, we can't see, that is the drones. So in females, in the queen, it is used for the oviposition, and in the worker bees, it can be used for the protection of the colony, that is the defense purpose. So the drones can't sting, that is statement is true, and royal jelly is secreted by the honeybee. This statement is also true. So here both statement 1 and 2 are true. And while coming back to third question, here also again statement type of questions, and the statement 1, the female of lac insect has true eyes, legs and wings. And statement 2, the tussar silkworm feeds on the castor. So if you could know the biology of the lac insect, the female insect is sedentary. After the first molt, it will loses all its appendages like eyes, legs and wings. And then it becomes sedentary on the host plant. So it is wrong. Only male possess all these things. Okay, male possess, and the tussar silkworm feeds on the castor is wrong. The silkworm that feeds on the castor is eri silkworm, and the tussar silkworm will feeds on the Terminalia arjuna and tomentosa. So this statement is also wrong. So here both the statement 1 and 2 are false. Yes, we can see, and the male possesses legs and wings explanation. And here in the tassar silkworm feeds on the forest trees like Terminalia arjuna and tomentosa, that thing. And the castor is a food for hairy silkworm. So here we are with one more matching. So there they have given the disease of honeybees, and here they gave the causal organism. So here we can see they have given four diseases, that is Nosema disease, Isle of Wight disease, then American foul brood and European foul brood. And here they gave the scientific names or the causal organism, that is Acaripsis woodi and Streptococcus species and Bacillus pluton and Nosema species. Okay, somebody is responding. Yes, for this answer is yes. Students, I am waiting for your response. You know this. Yes, gaming confidently, and Anchal and Dev Patel and Chaya, they have responded A or 1, that is true. So here Nosema disease is caused by, in Nosema, in apiculture, that is in honeybees, it is caused by the Nosema apis. And in case of silkworm, it is Nosema bombycis. Okay, only the species varies. So here it is Nosema species. And Isle of Wight disease is Acaripsis woodi, it's a mite. Then after American foul brood, that is caused by Bacillus pluton, and European foul brood is caused by Streptococcus species. Here and here. So A4, B1, C3, D2. That is 1. Yes. Option 1 is correct. Mm. And the next question, the standard Bombix mori loose egg pack contains. So mainly you should know one thing, one disease free layings, that is 1 DFL, contains 400 eggs. 400 eggs, and the standard Bombix mori loose egg pack contains, and one egg pack contains 50 DFLs. So yes, some of the students respond Anchal Gupta and Sneha Vishwakarma. 3. Yes, they are correct. Option is 3. But the behind this is the reason. 1 DFL disease free layings is 400 eggs, and one egg pack contains 50 DLS. So that's why it is 50 into 400. That is nothing but 20,000 eggs. Yeah. And the next question is Flaceri disease of silkworm is caused by the, here we are having four options, that is virus, bacterium, fungus and nematode. Okay. And Flaceri disease, the bacterial disease is caused as Flaceri disease of silkworm. Okay. So the causal organism is bacteria here. Yes, Chaya responded it's 2 yes. Bacterium. And the next question is the length of silk thread in a cocoon of Bombyx mori ranges from. Usually it ranges from 1000 to 1200 meters like that, but here appropriate option is 900 to 1000 meters is the appropriate one. And the next one, here also we have the disease names and the agent, the causal agent here. So first here we can see the Grasserie, Muscardine, Flecheria and Pebrine disease. And here the causal organism Nosema bacteria, and it is BM-NPV is Bombyx mori NPV. Okay, and fungus. So here we need to connect this. So Grasserie disease is caused by NPV. Then Muscardine disease, we all know it's a fungal disease. So B is 4. Then after Flecheria is caused by bacteria, and Pebrine disease is caused by Nosema bombycis. So here we are having A3, B4, then after C2, D1. A3, B4, C2, D1. Yes, option 2 is the answer. Yes, some of the students responded good, it's B. Here we will discuss the silkworm diseases. I like a flow chart. So then previous slide there was no space, so here I am mentioning it. Yes, ninth answer is 1, but here we were discussing the silkworm disease in a complete flowchart manner, so which will help you for easy understanding and easy remembrance. Protozoan, bacterial, and Graciela. Okay. So here the causal organisms in the pebrine disease or the protozoan disease is Nosema bombycis. So usually this disease is also called as a pepper disease or corpuscle disease. So you can remember it as a black pepper disease. Black pepper disease or corpuscle disease. Why? Because on the larva and even on the pupa, on the adult insects, we can see the black spots on the different regions, mainly on the abdominal regions. So it is called as a black pepper disease, which is caused by the Nosema bombycis, a Microsporidium protozoa. Then after in fungal disease, we have the muscardine disease, that is white muscardine fungi. then green muscardine fungi, then what else? And brown we are having, and yellow. Last one is yellow. Okay. White, we all know the causal organisms is Beauveria bassiana. So green muscardine fungi is Metarrhizum anisopliae. This also we all know. So brown muscardine fungi is Aspergillus. So Aspergillus flavus specifically. Okay. Then yellow muscardine fungus is Peciliomyces. Peciliomyces farinosus. Okay. And this is the fungal disease is called as muscardine, and the white muscardine fungi is Beauveria bassiana, and green muscardine fungi is Metarrhizum anisopliae. Brown muscardine fungi is Aspergillus flavus, then yellow muscardine fungi is Peciliomyces farinosus. And while coming to bacterial disease, here we are having the three again. And in the bacterial disease, which cause the damage to the digestive system, and another one is bacterial toxicosis, and the third one is septicemia. Okay. Here the causal organisms is streptococci. Choliaerogenous bacilli. These are the bacterial agents that cause damage to the digestive system. Then again, while coming to the second category, we were having bacterial toxicosis. So this disease is specifically called as Soto disease, that is caused by the Bacillus thuringiensis. Or BT, Bacillus thuringiensis. Okay. Then the last one is we are having septicemia. And here the causal organisms is streptococci and staphylococci. Streptococci and staphylococci, that is the bacterial disease is called as a flacheri, and in that their bacterial disease that cause to digestive system, then after the bacterial toxicosis and septicemia. So digestive system, we are having the causal organism streptococci, Proteus group bacilli, and choliaerogenous bacilli. Bacterial toxicosis is specifically called as Soto disease, that is caused by BT. Okay. Then after the septicemia, streptococci and staphylococci is there. And for the viral disease, here it is a big classification. It is sufficient for this. So if possible, you can take screenshot up to this. Then after, I will write the viral disease. Okay. So 1 minute I will move side. You take screenshot or either note down. So here due to sufficient space is not there, we will classify viral disease again. Here, the silkworm viral disease can be classified into two again, the occluded and inclusion type, then after the non-occluded or non-inclusion type. In that, okay, it's DV, densonucleosis virus. So silkworm disease 2, occluded and non-occluded. In occluded 2 and again in non-occluded 2. In occluded or inclusion type, we are having the NPV and CPV, that is nucleus polyhedrosis virus. Then after the cytoplasmic polyhedrosis virus, and in that in non-occluded infectious flagellaria and the densonucleosis virus, that is labeled as a DV. So here the families were bit important. Just you concentrate here. Here this virus belongs to Bacilloviridae. Okay, and this disease, by the disease that is caused by NPV, is called as some student said gaming confidently lists the viral disease. That's what I'm doing here. Borreliana virus. Then after CPV, that is cytoplasmic polyhedrosis virus, belongs to the family Rheoviridae, that is caused by Smithia virus. Okay, Smithia virus. So here infectious flagellaria is caused by mortar virus. Mortar virus. Mortar virus, and here it is caused by parvovirus. Parvovirus that belongs to the family Par. So these are the different viral agents. Okay. So NPV that belongs to the family Bacilloviridae and CPV that belongs to the family Rheoviridae, and that infectious flagellaria belongs to Iflaviridae. Okay, and densonucleosis virus belongs to the family Parvoviridae, that is caused by the parvovirus. And in while coming to that densonucleosis virus, there is one interesting thing. Actually, there is one disease in this. Yes. Okay. So complete silkworm disease I have discussed. First the bacterial agents, then after the viral agents, then after protozoan agents and fungal agents. And you know the febrile disease is caused by the protozoa Nosema bombycis, which is called as a black pepper disease or copsicle disease due to the peculiar symptom of black spots on the abdomen. Okay, even on the adult you can see, on the larva you can see, and even the lateral sides of pupa also you can see the symptom. So that is the peculiar symptom. And then next, coming to the fungal disease, the usually fungal disease is called as a muscardine, and we are having the 4 different agents. So like white muscardine fungi, then green muscardine fungi, yellow muscardine fungi, then brown muscardine fungi. So previously that was erased, so if you could be big slide it could be better. So green muscardine fungi, Metarhizium anisopliae, and brown muscardine fungi, that is Aspergillus flavus. And then after yellow muscardine fungi, Speciliomyces. Then after white muscardine is Beauveria bassiana. You all know this fungal classification. Then after the bacterial disease, that is called as a Flecheri. And Flecheri is having the three again. The bacteria that causes damage to the digestive system, and the bacterial toxicosis, then after the septicemial condition. Already we were discussing in the microbial control in IPM. So if you forget it, you can refer that. Then after the in digestive system, the agents are streptococci, then after Proteus, aerogenous bacilli and all. Then after bacterial toxicosis, specifically it is called as a Soto disease, which is caused by the Bacillus thuringiensis, BT. Then after septicemia is condition is caused by the streptococci, then after the staphylococci. This is all the classification. Apart from this, we are having the silkworm viral disease specifically. Okay, here you can see the viral disease. Again, we can divide it into occluded and inclusion type, and non-occluded and or non-inclusion type we can do. Okay, and finally by coming to occluded, you all know this classification here, NPV and CPV. And NPV belongs to the family Baculoviridae, and CPV belongs to the family Rheoviridae. These families are bit important. Okay, and the disease that is caused by NPV is usually called as Graseri, jaundice, or hanging disease. That is nothing but Weifel crank heat, and it is caused by the Borreliana virus specifically. And here the CPV that belongs to the family Rheoviridae, here the causal virus is Smithia virus. Okay. Then after the non-occluded or non-inclusion type, it is infectious Flecheri. Then after the other one is densonucleosis virus, that will usually abbreviated as DV. So here the causal virus is mortar virus. There the causal virus is parvovirus. And in that, if the disease, the virus and the bacteria, that is Streptococcus bombycis is there, that disease is called as Luceti or Clarity disease. Okay. This is this much. So it could be better if you take screenshots, or it could be much better if you write in your notes or somewhere else, so that if you could remember this classification, mostly you can answer these questions without any confusion. Okay. So let us see who keeps some messages, replies. Thanks, sir section. Okay. So now in the mulberry, the silkworm graseri is caused by BMNPV, that is Bombyx mori NPV. Okay. And here one more thing is NPV is also known as allothone. So this word you can say in the triple E. Okay. So while coming to the next question, that is the primary host plant of Antheria assamensis, that is Antheria assamensis is Moga silkworm. Okay. So here we are having the Prussia bombicina, Litacia monoptela, Prussia bombicina, Prussia americana, Prussia bombicina, Litacia cubeba, and Prussia bombicina, Litacia citrata. Okay. So will anyone answer this? Next question. Let me wait for 1 minute. Primary host plant of Antheria assamensis. So it is important the host plant scientific names you should remember. So one person responded, that is Chaya, kept A. Good. So Anchal also kept A. Then after gaming confident A. Yes, it's A. But how? That is the thing. You should know the common names of these things. So if any person knows the common names, you can write in the chat box. How you people are telling it's A? Yes, I am waiting for your answer. Yes, Som and Solu or Soalu. Yes, that's the local names. So Prussia bombicina is Som. This Letitia monoptella is Soalu. Okay, and this Prussia americana is avocado scientific name. Okay, this thing. Avocado scientific name. Then after this Letitia cubeba or Letitia citrata are generally synonyms. Okay, so the both are same. Good. Few students have answered it. And one more thing is that in tassar we will have the tropical tassar, Chinese tassar, and Japanese tassar. Then after Indian temperate tassar, and their specific scientific names are much important. Okay. So here you could see that tropical tassar is Antheria millata, then Chinese tassar is Prole, then like that Peruni, Yamame is from Japan, and Peruni is from China, and Prole is the tropical tassar. Okay. So and their hosts are important. So once go through it, then after in the 11th one. While coming to the next question, the 11th one is the identification symptoms for pebrine disease in 4th and the 5th install larva of mulberry silkworm is. So I said pebrine disease is caused by the protozoa which is called as a pepper disease or copsicle disease. So usually the color of the spots will be black color. Okay. So here the answer is black spots over the body. See, here in the images we can see the different black spots. So same like this we can see on the pupa and even the adult. Then the next question, the honeybee worker that always attend the queen is. So don't need to be confused. Usually this point you can't say anywhere. But if you could see with normally, here they have gave the 4 options. One is fanner bee, then after guard bee, retinue bee, and cleaner bee for this. So by the name itself, this it will suggest the duty what they will do. So here the fanner bee will maintain the temperature of hive. Okay, and here guard bee guards or protects the hive. Okay, guards the hive from predators or any other second colony bees. Okay, then after cleaner bee, so cleaning generally. So the leftover option is retinue bee. And usually what this retinue bee does is along with the queen there will be one group, and that group is called as retinue bee, usually of 7 to 8 bees. They will pick the pheromone that was released by the queen. Okay, that pheromone is distributed all throughout the colony. That's why queen is able to maintain it. If the queen is not able to producing that pheromone, the colony collapse disorder we can see. Okay. So here option is retinue B. This is the thing. Hmm. Again, one more matching they gave. Here they are asking about the wingspan and about their dimensions in inches. Okay, here we can see the wingspan of Hercules moth, Atlas moth, mulberry silkworm, and Tassar silkworm. And here on the other side, we can see the dimensions up to 12 inches, 2 inches, 6 inches, and 14 inches. Okay. So somebody is texting. So Hercules moth is the largest moth in Asia. Yes, some students kept 2 and some students kept 3. Yes, Dev Patel kept correct, it's 3. Okay. The wingspan of Hercules moth is the 14 inches. Yes, now also many students responding. Good. So it is the largest moth in Australia. Just moth in Australia. Okay. One thing. And Atlas moth, you know, this is the largest moth in our Indian context. Okay. Largest moth in India. So this Hercules moth is somewhat bigger than the Atlas moth. So here it can be the wingspan of 14 inches. Then after the Atlas moth is 12 inches. Then after wingspan of mulberry silkworm is 6 inches. Then Tassar one is 2. So A4, B1, C3, D2, that is 3. That's correct. Many of the students gave correct option like Dev Patel, Sneha. Then yes. Then after the 14th question is the Morus alba is the predominant food plant of which one? Direct. Morus mori. It's Bombyx mori, that is mulberry silkworm. Okay. And these all are Tassar silkworm. Okay. The term grainage in sericulture refers to disease-free and quality egg production, disease-free and quality cocoon production, disease-free silkworm rearing, safe storage of host plants seeds. So here the set of options they give. Then after that particular set, we have to need the select the appropriate option. Yes, usually the grainage definition tells that disease free and quality egg production. But there the work is usually also included with the sex determination, sex separation, then after the moth rearing and all. So it's nothing but it also includes the disease free and quality cocoon production. Okay. If both the options were there, you can go for egg and cocoon production. So here the option will be A and B, that is 1. Yes, students kept A. So this is the lag culture. Question from lag culture. Here they have given that host Aghani, Jetwi, Khatki and Baisakhi and their durations from when they starts and when they ends. So for that you need to see this. So here we can see there are two strains in the lac. One is Kusmi strain and one is Rangini strain. And in that the host varies. Mainly burr. Here the host plants are burr, then after semi-alta like that. And here we can see the not on the kusum, but the rest of the trees. And here we can we can see the Jetvi and Aghani crop. In Jetvi, the inoculation time was Jan and it ends by July itself. So if you could compare from these tables, we will go back. So it's a bit important when the crop starts and when the we will harvest it. So you need to focus here. So usually Aghani crop starts in the June to Feb and it ends by Feb. Then Jetwe crop, it starts in June and ends in November. Sorry, starts in Jan and July. Jan and July. So 6 months duration will be there in between Aghane and Jetvi. So once like this. Then Katki, it starts in the June and ends in the November. Then after Baisakhi strain, it starts in the October and ends in the July. So here A4, A4, then after where it went, B3, then C2, D1. A4, B3, C2, D1. Yes, 4. 4 is the option. Anybody gave correct answer? Many students kept 3, but it's actually 4. So for this, you need to revise the flow chart once again. Okay. It's not 3, it's 4. And Sneha, you gave wrong answer. It's 1. Not it's 1. C3 due to 1. Both 1 and 4 options are same only. 4, 3, 2, 1. So anything. And here we can see the one more matching, that is the green onion bulb, tamarind, mulberry and honeybee, and their associative things like tukra, beetle, nosimosis and scallions. Scallions are nothing but spring onions. Okay. So A will be comes to 4 directly. Then tamarind, already we were discussed internal feeder, primary pest and internal feeder, that is tamarind beetle. Then after mulberry we can see the tukra disease and honeybee osmosis, Nosema apis. So A4, then after B2, C1, D3. Okay. And the next question, the oozy fly in a silkworm is, here they have given the 4 options again: larval parasitoid, larval pupal parasitoid, pre-pupal and pupal parasitoid and adult parasitoid. Yes, anyone? Anyone knows this answer? Some Logeshwari Mohan, that is, she is telling it's 1. Chaya A. Specifically, the oozy fly is a endolarval parasitoid. So it's a larval parasitoid. So sometimes the question may ask it's either ecto or endo. So specifically it's a endolarval parasitoid. Endolarval parasitoid. Okay. Oozy fly causing damage. Then yeah. So here we are having the larval endoparasite. One boy responded good. And the next question is again statement wise question. In statement 1, the pollen basket in honeybee worker is situated on femur and the pollen basket in honeybee worker is situated on the tibia. So this is nothing but leg modifications in honeybee. Okay. So in honeybee, there were 3 modifications for 3 different legs. Okay, and one is for antennal cleaning, one is for wax picking, and one is for the pollen basket. So where I need to write? Ah, 4 legs. Hind legs. One for antennal cleaning, one is for wax picking, and one acts as a pollen basket. Okay, and 3 different legs are having 3 different modifications. Okay. So while coming to the pollen basket, we are having the on tibia, we are having 5 different parts in the leg, that is coxa, trochanter, femur, tibia, tarsi, you all know. And in that specifically on tibia, there is a one groove, like one groove kind, one groove like structure, and that groove acts as a pollen basket. Okay, and this pollen basket is also called as a corbicula. Pollen basket or corbicula. Okay, so this is present on the tibia, not on the femur. So the pollen basket in honeybee worker is situated on femur is wrong, and the pollen basket in honeybee worker is situated on tibia. It's correct. So statement 1 is incorrect. Statement 2 is correct. Which option? Yeah, option 3. So here we can see the 3 different leg modifications of the honeybee. So for the wax picking, we were having on the tibia, one spur will be there, antennal cleaning will be there on the foreleg, and hind leg pollen one. And the next question, while coming to, yes, someone responded at set 3. Many students were responded for this. Okay. Then while coming to next question, given below are 2 statements, that is statement 1, shellac is a resin secreted by the female of lac insect on the trees in the forest. Yes, it's a resin secreted by the lac insect. This is correct statement. Then after statement 2, shellac is composed of aluritic acid, malic acid, and shelloic acid. So here usually malic acid is present in case of apple or other any remaining fruits. So malic acid that won't be present in shellac. So due to this single acid, this statement goes wrong. So you need to be read very carefully. So usually in overflow, what students will do, shellac is composed of aluritic acid, malic acid, and shelloic acid. You goes for both the statements were correct, but it's not. Okay, malic acid is not present in the shellac. Okay, so state this is wrong. So statement 1 is correct and statement 2 is incorrect. Yes, many students responded C or 3 yes. So reading questions very carefully is very much important in attempting the answers. And while coming to the next question, here we can see the matching oak tassar silkworm, muga silkworm, then after mulberry silkworm and tropical tassar silkworm. Here we can see the different states, that is Jharkhand, Uttarakhand, Assam and Karnataka. Nothing but they were asking the production from which state is highest. Yes, many students responding. I'm waiting for your response. Yes, Nisha, C or 3, yes, it's correct. Sneha Chaya also responded, it's 3. What 3 we need to? So oak tassar silkworm is highest in case of Uttarakhand. Then Muga silkworm is specific to Assam. So that's why the name itself gives the Assamensis. Okay, and mulberry silk from silk worm from Karnataka. Then tropical tussar, the leftover option is Jharkhand. Simple. So A2, B3, C4, and D1. A2, B3, C2, D1. Yes. 21, option 3. So the predatory fauna of lac insect in India is, so they were giving many insect names, but in that Eublema amabilis and Holocera pulveria. One belongs to the Noctuidae family and one belongs to the Gellichidae family. Okay. Noctuidae member, then after Gellichidae. So I'm responding for students. So here the predatory fauna is A, Eublema and Holocera, that is A only. Holocera, yes. And while coming to next question, the Athalia lugens belong to which of the following insect order? Athalia lugens proxima. Yes, students responded. Anchal Gupta, Chaya A. For the next question. Yes, I'm waiting for your response, students. Anchal Gupta, Chaya, then Sam, then Nisha. All were responding. It's 4. Yes, Athalia lugens proxima is 1. Until now, it's a known, only single known pest from Hymenoptera. Okay, that belongs to the family Tenthinidae. Hymenoptera. Hymenoptera. Next, the 24th question is India is known for the rich fauna of the serigenous insects due to. So, Samia ricini, Attacus atlas, Bombix mori, Aticius selene, then after Philosoma ricini, Antheria maileta, Antheria prolei, and Friti. So, all these insects, the silkworm insects, can be seen in our India fauna. Okay, so the options goes to A, B, C, D. A, B, C, D. Some students responded Sneha and Nisha. Yes, it's 4. Given below are two statements, and the statement one is lac cut from the host plant is called as a stick lac, and lac used for the manufacturing purpose harvested after the larval swarming occurred is called as a punky lock. Yes. Yes, Anchal Gupta A. Yes, only Anchal Gupta responded it's A. So stick lac or punky lac or punky lac, both are same. Okay. So after the nymph emergence, the leftover one is called as a punky lac. So mainly that is the base we will use for the everything. So here statement 1 and statement 2 are the correct. Yes, now many students responded. Nisha and Sam also kept A, Chaya A. Wax glands in honeybee present on the 4 to 7 abdominal sterna, that is on the ventral part. Then after the 4 to 7 abdominal terga on the dorsal part. Then after the 4 to 7 abdominal pleura, that is on the lateral sides, and 8 to 11 abdominal sterna. So knowing the segments is not enough. The exact position, whether it's on the lateral sides or on the dorsal side or it is on the ventral side, we need to know. So wax glands are present on honeybee in the 4 to 7 abdominal sterna, that is on the ventral side. Okay, so option 1 is correct here. So here only A. So option 2 is correct. Yes, now Sam responded it's base Neha 2. Then Chaya also B. Good, many students were responding today. So these are the wax glands. An endemic honeybee which was discovered in the Western Ghats after a gap of more than 200 years is. So they have given the different epispecies. In that they were asking which was discovered from the Western Ghats. Apis caryngodion and Apis carjodion. So here either one or two can be correct. So both the options were correct in this case. Ah. And CRC stands for Chowki Rearing Committee, Chowki Rearing Capability, Chowki Rearing Center, and Chowki Rearing Culture. Yes, CRC. Nisha responded one. Sorry, Nisha, it's wrong. It is Chowki Rearing Center, not the committee. The full form of CRC is Chowki Rearing Center. So, Oak Vibe and Palam Mishra. Yes, she responded correct. So, Chowki Rearing Center. Then after, while coming to the next question, here also we can see the two statements. The statement one is Megatriosa hirsuta is a common insect pest of Terminalia arjuna, and statement 2 is Megatriosa hirsuta is a common insect pest of Morus alba. So you should know what is now this Megatriosa hirsuta. Then you can be answering this question. Okay. Actually, this Megatriosa hirsuta is jumping plant lice. Or silid simply. Okay, it's a pest of forest crops. So in this case, this statement becomes correct and this statement becomes wrong. Okay, it's not the pest of Morus alba. So here the statement 1 is correct and statement 2 is incorrect. So option 3. Then after, while coming to the next question, again statement type of questions. Yes, students responded 3. While moving to the next question, statement 1, shellac is resin secreted by the female Laccifer lacca, that is scientific name of lac insect, on trees in the forest. Yes. And the statement 2, shellac is made up of gum and arabic and other natural waxes. Yes. Chaya ye Mahim Mahendra C, Nisha 1, Mishra C. Here the statement 1 is only correct and statement 2 is wrong. So statement 1 is correct and statement 2 is incorrect. Option 3 is correct. Some only few students responded for this question. So common food plants of Laccifer are Palas, Burr, Babul, PGNP is one set of option, and the second set is Kusum, Palas, Burr and Babul. And third one is Care, Gooseberry, Mulberry, and here D, Citrus, Avocado, Neem and Guava. So Laccifer is a lac insect which can be grown on the forest host plants. Mainly specifically we are having the Kusum strain and Rangini strain. According to that, only these two sets, the option A and option B are its host plants. So here A and B are correct. And Chowki silkworm rearing under nylon nets is performed for. Yes. No, it's not done for Antheria prolei. Somebody kept option 1. No. Chowki silkworm rearing under nylon nets is performed for Antheria mylitta. Okay. Indian tropical tassar. So here one interesting thing and why they are raising under nylon net conditions is simple, to prevent from the pest. Okay, to give protection from the predators, they were raising under the nylon nets. And the next one, sting of honeybees consists of acid gland, alkaline gland, labial gland, hypopharyngeal gland. So we know sting is the abdominal part, the last part. So directly you can eliminate these two options. Labial and the hypopharyngeal glands usually located in the head portion. So the rest over things are acid gland and alkaline gland. That's it. A and B. So this is the sting apparatus structure. Here we are having the acid glands. Then after venom sac, here we are having the alkaline gland and the sting sheath. Then after the final, the pointing. So green muscardine in silkworm is caused by, they are giving here also 4 options. One is BMNPV, that is Bombyx mori NPV. Then after Beauveria bassiana, that is, and the third one is Numeraria rilei and Nosema bombycis. We know here then I have named itself is a green muscardine, which is a fungi. We know this. So this one won't becomes, Nosema bombycis won't becomes. Then we know Beauveria bassiana is a white muscardine fungi and the Numeraria rilei. Actually, it was a previous name of Metarrhizum anisopliae. Okay, so 3. Then after, while coming to next question, Bacillus thuringiensis BT was first isolated from a deceased silkworm by Ishivata. Sam responded and Mahendra also responded. It's B or 2. It is discovered by Ishivata. Okay, that only we are calling it as a Soto disease. Soto disease. And next, the royal jelly is secreted by the either maxillary glands, labial glands, hypopharyngeal glands, and mandibular glands. Yes. Yes, many students responded Sneha and Chaya's 3 or C. Yes, it's correct. Royal jelly is secreted by the hypopharyngeal glands. In worker bees, when the second stage, in the second week of the development, that is from day 7 to 14. Okay. Secrets royal jelly. So here royal jelly is secreted by the hypopharyngeal glands. Many students responded here. Yes, it's 3. Answer is 3, but I was discussing the other points from the royal jelly or the hypopharyngeal glands. So, okay. Actual royal jelly is called as a bee silk. Okay. Then after in the workers development from the day 7 to 14, this specific period, this 1 week time period, worker bees will secrete the royal jelly. And specifically that period bees is called as a feeder bees. Okay, and this hypopharyngeal glands is well developed in case of workers and vestigial or non-functional in case of queen, and it's completely absent in case of drones. Okay, absent in drones, vestigial in queen, and well developed in the workers, and these bees are called as a feeder bees. From day 7 to 14 they secretes royal jelly. Royal jelly is also called as a bee silk. Given below, or and the next question, given below are two statements, and the statement one is mulberry silkworm is monophagous insect that feeds only on Mora species, and statement 2, eri silkworm is another insect that feeds only on the Ricinus communis, that is the scientific name of the castor. Okay. So we all know mulberry silkworm is only consumes or there can be raised on the mulberry plant. So it is true. So mulberry silkworm is a monophagous insect that feeds only on the Mora species, statement is true. And this eri silkworm, it feeds on the Ricinus communis, that is castor plant, but it is a polyphagous insect actually. It can be raised on many plants. Okay. So here they gave that feeds only on the Ricinus communis is wrong. It can be feed. Actually, it's a polyphagous. So this statement becomes wrong and this statement becomes correct. So statement 1 is correct and statement 2 is incorrect in case of this question. So yes, some students responded C, only Mahendra gave the correct option C. And while coming to the next question, the royal jelly on which queen larvae are fed contains, it's a chemical name. Okay, IPAC name. That is, they are giving 10 hydroxy trans 2 decanoic acid, 10 hydroxy cis decanoic acid, 10 hydroxy cis 4 decanoic acid, and 10 hydroxy trans 4 decanoic acid. That's what you need to remember, the chemical names also. Somewhat important thing. If you could get this such questions, you will be able to answer. Actually, see how this was confusing. The trans, opposite and cis, the same side, 2,2-decanoic acid, and here they were changing the 4-decanoic acid. So actually the royal jelly is 10-hydroxy trans 2-decanoic acid. 10-hydroxy trans 2-decanoic acid. Okay. Pertinent question. Only gaming, Nisha and Chayef, this all kept A. Then the next one is headquarter of AGRIP on honeybees and the pollinators is situated in Punjab Agriculture University, Ludhiana, IIR, New Delhi, Hisar and Bangalore. Anyone? AGRIP of headquarter of AGRIP on honeybees and pollinators is situated at Sam kept 2 and Mishra kept C and gaming confidently kept B. I got wide variation in the answers. So actually this headquarters is present in case of Indian Agriculture Research Institute, IARA, New Delhi. Okay, AGRIP headquarters were present in the IARA, New Delhi. So while coming to the next question, what is grainage? This was asked in ICAR JRF 2023 Shift 2 paper. They have given 4 options here: where disease free and quality silkworm eggs are produced, where disease free and quality silkworm cocoon seeds are produced, where disease free and quality silkworm rearing conducted, and where disease free and quality silk is produced. So I said earlier, actually the grainage definition tells that itself. The place where production of free and quality silkworm eggs are produced. But the grainage place also includes other operations such as sex separation and moth rearing and moth emergence and all. So in that it also includes the cocoon production also. But specifically when 2 options were given like this different, you need to go for the place where eggs are produced. And if in case like in the previous questions we have seen both eggs and quality cocoon production is there, such options, 2 options were combined. In that case you can go for 2 options. Otherwise, when single option is there, you need to give a priority to free and quality silkworm eggs are produced. First one. Okay. While coming to next question, given below are 2 statements, again statement type of questions. Statement 1, for commercial purpose, Mora species plants are propagated only through seeds. Normally, mulberry plant is propagated through cuttings, grafting and layering. So here, well, I got one message in chat box, that is, sir, in IARA, sessional and final exams are easy or hard in entomology. My dear Scirpophaga incertulus, please be focused on the particular JRF exam. Okay. Then after when you come to IRA, at that time we can discuss. However, when you come, I will stay in IRA itself. At that time we can see. Okay. And for commercial purpose, more as species plants are propagated only through seeds. For commercial purpose, it won't be recommended. Only for academic research purpose, they will do. This statement is wrong, not for commercial purpose. Okay, for only research. You can think. Okay, and statement 2, normally mulberry plant is propagated through cuttings, grafting and layering. Okay. So here statement 1 is incorrect, but statement 2 is correct. The common name of recently discovered honeybee from Western Ghats is. Already we were discussing that previously, which was founded out after 200 years gap, that is Apis caryngodion. That is nothing but Indian black honeybee. While coming to the next question, again here also same the statement type of questions. Given below are two statements. Statement 1, volatinism in insects is number of generations produced in a year or per year. Yes, volatinism is number of insect generations produced per year. And Morus albi is a perennial tree in northern India or northern India. That is also true. Here both statements were true or correct. 44 is A. And again here while coming to the next question, we can see the here also again two statements. One is Tukra disease in mulberry is caused by whitefly is the first statement, and shellac is a resin secreted by the female lac insect on the host plant. So anyone? We will wait for 1 minute. How Tukra disease in mulberry is caused by whitefly? It is caused by mealybug, Maconellicoccus hirsutus. Okay, and that name will be different from while considering the rest of the mealybugs. So Maconellicoccus hirsutus is the mealybug that causes Tukra disease in mulberry. Okay. Then after shellac is a resin secreted by the female lac insect on the host plant. Yes, some Mahendra, Tukra disease mealybug says it's correct. That's what I'm telling. And while coming to the next, shellac is a resin secreted by the female lac insect on the host plant. Yes, Chaya, it's caused by mealybug. And here the statement 1 is wrong, but statement 2 is correct. While coming to the next one, oozy fly is a serious pest of. We have discussed already. Yes, oozy fly is a pest of. Yes, Mahendra, Sam, Mishra, Anchal Gupta responded. It's 2 and Chaya 2. Oozy fly is a pest of silkworm. Okay, endolarval parasitoid. Since here silkworm is a beneficial insect, we were considering it as a pest. Otherwise, it's a parasitoid. Okay, and same again. Previous also this question we have seen. Oak tussar silkworm is more prominent in case of Uttarakhand, and Muga silkworm is more prominent in case of Assam. Mulberry silkworm is more prominent in Karnataka, and tropical tussar from Jharkhand. So, is there A2, B3, C4, D1. A2, B3, C4, D1. Yes. Third option. Okay. Next. The size in of a healthy adult Lachfer laka ranges between. Usually such questions no student will remember, even I will not remember. So if you could read triply correctly, they have mentioned one point there. The nymphal size is 0.6 mm. So adult will be somewhat more than that. So here the appropriate option or the correct option is 0.6 to 1.5 mm. Okay. And some students responded B, 0.6 to 1.5 mm. Okay. Nymphal size is exactly 0.6 mm and the adult will be somewhat more than that. So that range is 0.6 to 1.5 mm we can go. And usually the life stages of the male Lachfer laka consist of egg, larva, pupa, adult, all the stages it will have. So here A, B, C, D. Which of the following countries is the largest producer of lac? Actually, lac is indigenous to India itself. Okay, so the largest producer is India. Option 2. So it is indigenous to India. Given below are two statements. Statement 1 is the oozy fly is a larval parasitoid of the silkworm and the hide beetle, that is Dermastis species, feed on the silk, pupa and cocoons. So here it is from the pest of the silkworm. Okay, oozy fly we know it's a endolarval parasitoid of the silkworms. So statement 1 will become correct, and hide beetle, Dermastid beetle usually feed on the silk, pupa and even the cocoons completely. So both the statements are correct in this case. So many students responded. Mishra, Anchal Gupta, Chaya and Nisha kept 1. Yes, it's correct, students. And while coming to the next question, here also again statement type of questions. Bombyx mori is solely domesticated worm throughout the world. Is the first statement. And next one is Bombyx mori is solely domesticated worm in the India. So here you need to understand the meaning correctly. So Bombyx mori, the mulberry silkworm, can be domesticated irrespective of the countries or any in climatic conditions. Mostly it will be in the artificial conditions while rearing. So it can be domesticated easily. So the statement 1 is correct. And Bombyx mori solely domesticated worm in India. So when compared to other silkworms like tassar or eri silkworm or muga silkworm, okay, it is the one we were domesticating it or we were raising it. Okay, so both the statements were correct. Bombyx mori is solely domesticated worm throughout the world. It's also correct. And Bombyx mori is solely domesticated worm in India. So this statement also correct. So both statement 1 and statement 2 are correct. Hmm. While coming to next question, average honey in cages produced by Apis dorsata, that is rock bee, per hive is more than the Apis mellifera. Then average honey in cages produced by Apis laboriosa is slightly higher than the Apis dorsata. So Apis dorsata, rock bee, Apis mellifera, you know, and average honey produced by laboriosa per hive is slightly higher than the Apis dorsata. So when my first session in the PYQ in 2025 paper, when I have attempted, somewhere I have read it per hive ranges of this. Okay. Usually average honey kg produced by Apis dorsata per hive will be 37 kgs. Is more, and in case of Apis mellifera, it is 15 to 20. 15 to 20 kgs. And statement average honey kg produced by Apis laboriosa is slightly higher than Dorset. So this Apis laboriosa is a important pollinator in apple crop. Okay. Apple pollinator, and here Dorsetta, and here it produces 60 kg. Okay, and slightly higher than the Dorsetta, again Dorsetta 37. So here also the both the statements are correct. Okay. 54. Which disease is caused by fungus Beauveria bassiana in silkworms? That is nothing but white muscardine fungi. Okay. It's a muscardine fungi. Specifically, this is a white muscardine fungi. White muscardine fungi. Yes. And rest of the pebrine, fletcheri, grasserie we have discussed already. Grasserie is done by caused by NPV, then pebrine is from a protozoa, fletcheri is from bacteria. Okay. So here it's a muscardine fungi, muscardine disease. Hmm. And the next question, it was given in the ICAR JRF 2025. That is, they have gave the threat, and on the other side they have gave the impact. Okay, here they have gave the reasons or threats, that is varroa mite, that is ectoparasite. Okay, Acaripes woodi, that causes Isle of Wight disease is endoparasite, and varroa mite is ectoparasite. Okay, and colony collapse disorder, neonicotinoids, deforestation or urbanization, and the climate change. Then here on the impact for this, here pesticides that harm the bee nervous system, parasite, sudden loss of worker bees, reduce pollen and nectar source, and habitat loss. Okay. Here varroa mite is a parasite. Specifically, it is a ectoparasite. Okay. And colony collapse disorder is the sudden loss of worker bees. B will be 3. And neonicotinoids, we know that is a pesticide. So option 1. Then deforestation or urbanization is nothing but habitat loss. Okay. And climate change causes reduced pollen and nectar source. So if climate changes, what will happen? Some of the plants may not be survivable. So there we can see the pollen and nectar source reduction. So here A goes to 2 and B goes to 3, then C1, D5 and E4. So which option comes? A2, B3, C1, D1, 4. Yes. So first one is the option. Okay. So with this, all our 54 questions completed. So very long session. Thank you students for your support, and in this class I was happy with your interaction. So many of the students were joined and many of the students were actively answering the questions in the chat box. So from these questions we could analyze something. Mainly the important one is that mostly they were concentrating on the causal organisms. So in case of beneficial insects, mainly the apiculture, sericulture or lag culture, concentrate on the pest and disease of those things. And if you could remember the silkworm diseases, like how I explained in the flowchart, it is very easy to understand. So rest of the symptoms you can read or remember, but while confusing point is which causal organism causes which disease and what that particular disease is called. That is important. So concentrate on that flowchart specifically, and in case of honeybees, go through the species, then after how they can be maintained and what are the pest and diseases. So all this beneficial insect portion you can read it from the Elements of Economic Entomology triple E book. So it is the well standard book, so you can read it from that. So one student is telling Saurabh Chand Yadav 1234, sir explain in Hindi. I can't speak that much fluent Hindi, so I will explain in the English itself. So I'm sorry for that. I will try to learn. That's it. Thank you, students. Bye.